I just have a few slides here, all right, because today we actually have some data that we can analyze, right? The past two experiments have all been observational data. This time we actually have numerical data, all right? So there's a lot of fun things that we can do with numerical data, and one of them is just running some statistical analyses on it. All right, so statistics, all right, I'm keeping this very, very broad because this is not a math class. This is not a full semester science class here. But statistics are used to state how unlikely our results are to reflect a chance process. All right, so basically we're looking at our data and saying, is this pattern really existing, or is this just due to chance? All right. It's important to remember that science can never prove something. Right? So if you're writing down that this has proven our hypothesis, right, that would be an incorrect statement, and I will likely make a comment on that. I'll be going through your gravity labs tonight, making comments for you. Because science only gives us the confidence in saying the likelihood of something. Right? So we're not proving something, but we're saying this is the likelihood that what we're seeing is actually real. All right? So if we look at this graph here, right, and we have different months and number of days of snow, right? Do they look different, right? The different months, do they have different days of snow? Yeah. Right? February is definitely the most snowy out of those 4 months there. Right? What is your confidence level in saying that that's not just due to random chance? You could be fairly confident, all right? That's a higher bar, all right? But you don't have anything to back up your confidence, right? So this is where we need to run stats to say that February, yes, it looks like we have a lot of snow in February, right? Is February the snowiest month? Maybe, all right? It looks likely, right? But I need some data to back that up, right? And this is where stats come into play, right? Especially when we think about sample sizes. Right, so typically, maybe this data here, we wanted to say February was the snowiest month on average, right? And we only used 2 years worth of data, right? Is that going to give us a likely, an accurate picture of what's going on, say, over 50 years? No, because we're just really using 2 years, right? Our sample size is very, very small. Right? Now, if we wanted to be more confident here, we could increase our sample size, use more years. All right? So in our experiment with our Martian data soil, how many replicants do we have? What is our sample size? How many groups do we have in the class? 6, yes. We have replicated this experiment 6 times. Right? So there's 6 pots of Martian soil, 6 pots of Martian soil with compost, so on and so forth. Right? So our sample size is 6. Right? Now, would we think that's a fairly large sample size? No, not really. All right. It's for this class, it's large enough for us to get some data and run some statistics on it. Right? But if I were to publish something in a paper where I really wanted to be confident in my results, right, I have a lot more than just 6 sample sizes. All right, so I'm going to move on to the next slide here. When we do stats, all right, we have to choose a probability of accepting or rejecting essentially our null hypothesis. All right, so can anybody remember what the null hypothesis is for this lab? I know you wrote it down like almost 2 weeks ago now.
It was something like there will be no difference in the height of the plants differing in soil type, something like that.
Yeah, exactly. Yeah, so there will be no difference between our treatments, right? All right, so we need to choose a probability of basically a numerical value that says what is our acceptance or rejection of that null hypothesis. All right, and so that makes sure that we do not make a mistake sampling variation in our natural biological variation that exists. All right, so for example, not every single one of those plants grew exactly the same because there's variation in nature, right? So we would set this threshold at 0.05. All right, so we would expect to see a difference by chance, right? Totally random, about 5% of the time. All right, so that means 95% of the time, all right, we should be fairly confident in saying yes, there is a pattern happening that is not due to chance. So that magical number in biology is what I like to call that, 0.05. All right, so how do we get this number? And how do we actually apply it to our data? All right, so we're going to be doing a test called an ANOVA. All right, so an ANOVA is going to look at the means of 3 or more groups. All right, so how many treatments did we have in our lab? We had 4, yeah. So we had 4, so we had 4 different groups, right? And because we have 4 groups, and we're going to compare the average growth rate between those 2 different soil types, right? We're going to use an ANOVA to analyze our data, right? This ANOVA is going to give us what's called a p-value, right? And this p-value goes back to that threshold we were just talking about of 0.05, right? It's going to inform us either to reject or fail to reject the null hypothesis, right? Now in science, this can be some confusing wording, right? Why don't we just say we're rejecting the null and accepting the alternative, right? This is just the terminology that scientists use, all right? So we're rejecting or failing to reject the null, again, because we can't prove something, right? So we're not proving the alternative. All right, so when we look at our p-values, again, we're going to look at that magic number of 0.05, right? If it's greater than 0.05, we will fail to reject the null, right? We're saying that what we are seeing is not due to chance. Excuse me, I said fail to reject. See, even the wording confuses me sometimes, right? If we're failing to reject the null, right, we're essentially keeping the null, right? Right? That means that the variation that we are seeing is due to random chance, right? So that's completely the opposite if we have that p-value less than 0.05, right? So in this case, we would be rejecting the null and saying that the pattern that we are seeing, right, is not due to chance. There is something going on there, right? So basically, we're using these p-values to give us the confidence in saying that the pattern we see is actually a pattern. It's not just some random thing that popped up. All right, any questions about the ANOVAs? I know I'm keeping it very simple here. If you have questions while doing it, please let me know, and I expect there to be questions. All right? I'm going to go through a couple more things that we'll be doing in lab today. That way we don't have any more lecture to go through, and we don't have to pause and start again. All right, so once we've analyzed our data, right, we want to graph our data so we can visually see the pattern that may or may not exist in our data. All right? So when we're graphing our data, we have to think about our variables. Right? And there's 2 types of variables that we can have. So one would be discrete. Right? So I like to think of different discrete categories, right? Blue, orange, green, yellow, right? They're very discrete. If something is yellow, it is not going to be blue, all right? So if we look at our continuous variables, right, we're not necessarily putting them in categories, right? This is where there's a huge range of possibilities that could be present, right? So for example, 1.1, 1.2, 1.3, 1.4, so on and so forth. Right? That would be a continuous range of values. Right? So when we're looking at our data, we want to be able to look at it and put them, say, is this a discrete value? Is this a continuous value? Right? Now a graph, the purpose of a graph, right, is to be able to look at your data, right, and show the reader what they are looking at without them having to read your entire report. Right? So if I just looked at the graph, I would have enough information to say, oh, this is what this graph is about. I get a sense for how they came about getting this data. All right? All right, so for this class, right, we'll be using a bar graph for the lab. Right? And we'll use a bar graph when our x variable is categorical. Right? So x being along the horizontal axis here. Right? So what are our categories for our lab?
Type of soil.
Types of soils, yeah, we have those 4 different soil types, yes. All right, so that's always going to be the independent variable. All right, now what type of data have we collected? Height. We've collected height data, yeah, and we're actually going to turn that height data into the average growth rate. All right, so our growth rate would be our y variable, and it's going to be continuous. All right, so our y variable is on the vertical axis there. It's a continuous number. Now one thing to note with these graphs, right, just like you've done with your photographs you've included on your other lab reports, right, we need to have a figure caption. right? And the same thing goes with these figure captions. I want to know what I'm looking at, right? I want to know what species I'm looking at. I want to know how many replicates are included. Give a very brief summary of the experiment, right? It can be very brief. A researcher is not going to try to replicate it from the sentence you included in your figure caption. All right, they would look into the methods for that. Right, brief summary of the experiment and brief summary of your results or any trend that you saw. All right. Now, one of the other elements to the graph are error bars. All right, so we know that data is variable. Right, there's going to be a little bit of error within our data here, and so we want to put that onto our graph. All right, and we're going to graph standard error. All right, so there's other ways that you could graph this variability, such as standard deviation. All right, I'm going to use standard error. I like standard error because it controls for the sample size. All right, so if we use different samples, for example, maybe if one of our plants didn't grow in a pot, and all of a sudden we had 5 replicates for one soil type and 6 for the rest, all right, we'd have different sample sizes. All right, standard error would be excellent in that. All right, so we're going to do standard error. And so if we look at that graph I have up there, right, we see we have our error bars. Right, it goes above the mean, so this line up here, that would be the mean. It goes above and below that mean. Right, it shows us the range of error that could be present. Right, now notice that one of those bars has a much larger error bar than the other. Right, so if you're looking at them, and this graph is looking at techniques to measure oxygen saturation in blood, right? And if I was looking at that and thinking, oh, which technique is the most accurate, or which one maybe should I use, right? I might lean towards the one that has the smallest error bar, right? Because it's closer to that mean every time I do it. All right, so we're going to include error bars on our graph, right? I've actually set up the document, so that's going to be automatically calculated for you. All right, it's just going to populate for you. All right, any questions about the graphing portion? All right, the last thing I'm going to ask you to include is a table, right? And this table is going to tell us our germination rate, right? So we recorded the days that it took to germinate, right? And I just want a nice little table to synthesize this information. Right, so I put this table up here with a bunch of boxes that kind of gives you an idea of what a table should look like. All right. The difference between a figure caption and a table caption, right? The table caption is going to go above the table, right? Whereas a figure caption is going to go below it. Right? The independent variable is always going to be in the far left column. So in this case, that would be age or years. Right? All the other columns are going to be labeled, right? If there's any units, include units, right? So our units for today, and you'll probably hear me repeat this several times. I don't think I actually put the units in our procedure, right? But essentially, we measured height, and what did we measure height in? Centimeters, yes. And I am getting growth rate data by dividing the number of days by that height. All right, so it'd be centimeters per day would be our units. All right. For units for germination, right, I'm just asking number of days until it germinated. All right, so your units there would be days. All right. What else here? Talk about it goes above. We label it the same way, right? Except it's saying figure, we say table 1. All right. It's going to have a very short figure caption again, right? And notice this table is very neat, right? There's not a lot of lines going on here, right? There's only 3 lines, right, and no vertical lines, right? So make it very nice and neat. Right? Too many lines just kind of distracts away from the point sometimes. So I have included these slides on Moodle so you can go back and reference them. All right. Any questions about anything we have talked about? Yes.
Okay, this may be a stupid question, but how are we creating said table and graph? Is there a specific way that we're going to do that?
Yes, and that is in the procedure document I have online. Okay. So the graph you'll be using Excel. The table, I said use Word. You can use Google Docs, right? I just don't like creating tables in Excel because I think it's hard to try to export them out of Excel. But yes, there's a couple different ways you can do it, and they are laid out in the document there. Any other questions? All right. So one quick side note before we transition into all these moving parts, right? you listened to that Guardians of the Galaxy episode, maybe you have yet to do it, right? So that assignment, I believe I put as due at midnight tonight, right? And she talked about these aggregates, right? So I have this pot of Martian soil that I potted as soon as I got the soil in to see how the water held inside of here. And then I let it dry out because I forgot about it, all right? But I came back to it, right? And it's very, has a lot of aggregates in it now, right? Because it's dried and clumped up. And this is essentially that same process that scientist was talking about in this podcast, right? She wanted to try to introduce aggregates, so she watered it, let it dry out, and then we had some aggregates instead of that very fine, dusty powder we started with. All right. I just thought that was cool. Thought I'd share that here. I'll leave it up front if you want to look at it. So, make the final measurements on your plants if you have not already. All right. Get those data sheets and have somebody input them into Google Sheets. Right. Follow the procedure that I have on Moodle. Right. That should give pretty much step-by-step directions. If I have missed something, let me know so I can correct it for the next time I teach this. All right. Once you are done with all those measurements, we don't need our plants anymore, all right? So we have to clean up our experiment. So in the lab, I have set up a few tubs, all right? There is a tub for the Martian soil, there is a tub for the Martian soil that had compost in it, and then there's a big bucket for all the other soil types, all right? I have them labeled in there, all right? I really want to keep the Martian soil and the Martian soil with compost separated out, all right? So I can use that in future labs here. The plants themselves, if you just want to pull them out of the soil and put them right into that blue bucket, right, that's awesome. And then the dirty pots, there's also another little tray that you can put your dirty pots in when you're done. All right, so cleanup should be really simple. Yeah, so with that, let's get started with our lab. Most of it we can do in here because it's a larger space, and I think it's better COVID-wise for transmission right now. But we can walk back and forth between the lab, whatever you need to do here. All right, so we're doing a lab, which is weird to do via Zoom. What are your lab first?
I don't have Excel works on my computer, so we'll just make it work. But, I have the Google Sheet open to put like the class data in. Yeah. So once in the classroom,
because most of it'll happen in the classroom.
Can she carry all of those? And really? Yeah, I think so. Because we need to like.
Are we bringing the plants down here?
You can. There's really no reason to bring them in the classroom once you got your data. But then you'll bring them in the lab eventually and take care of them.
We can't edit the sheets.
All right. Good call. That is an easy fix.
I say you just go.
All right, you should be able to edit it now.